Meiosis is a type of cell division that results in 4 daughter cells, each with half the number of chromosomes of the parent cell. Meiosis includes 2 consecutive cell divisions, meiosis 1 and meiosis 2. Prior to meiosis, the cell must go through interphase. The DNA in the cell is copied, resulting in 2 identical full sets of chromatins. Outside of the nucleus, the centrosome duplicates, and both resulting centrosomes contain a pair of centrioles. The chromatin within their chromosomes is uncondensed. Prophase 1 is the longest stage of meiosis. During prophase 1, the chromatin condenses into chromosomes, and they become thicker, shorter, and visible under a light microscope. The duplicated homologous chromosomes pair and crossing over occurs. Crossing over is the process that can give rise to genetic recombination. At this point, each homologous chromosome pair is visible as a tetrad, a tight grouping of 2 chromosomes, each consisting of 2 sister chromatids. The sites of crossing over are seen as crisscross non-sister chromatids and are called chiasmata. The nucleolus disappears during prophase 1. In the cytoplasm, the meiotic spindle, consisting of microtubules and other proteins, forms between the 2 pairs of centrioles as they migrate towards opposite poles of the cell. The nuclear membrane disappears at the end of prophase 1, allowing the spindle to enter the nuclear area. During metaphase 1, the law of independent assortment takes place. During this phase, the centrioles are seen at opposite poles of the cell. The pairs of homologous chromosomes become arranged on a plane equidistant from the poles called the metaphase plate. Spindle fibers from one pole of the cell attach to one chromosome of each pair, and spindle fibers from the opposite pole attach to the homologous chromosome. During anaphase 1, the law of segregation occurs. Anaphase 1 begins when 2 chromosomes of each tetrad separate and start moving towards opposite poles of the cell as a result of the action of the spindle apparatus. In anaphase 1, the sister chromatids remain attached at their centromeres and move together towards the poles. During telophase 1, the homologous chromosome pairs complete their migration to the 2 poles as a result of the action of the spindle. A haploid set of chromosomes is at each pole. A nuclear envelope reforms around each chromosome set. The spindle disappears, and cytokinesis follows. In animal cells, cytokinesis involves the formation of a cleavage furrow, resulting in the pinching of the cell into 2 cells. However, in plant cells, the cell plate forms to duplicate the cell. After cytokinesis, each of the progeny cells have a nucleus and a haploid set of replicated chromosomes. During prophase 2, the centrosomes duplicate, which occurs by the separation of the 2 members of the pair. The centrosome splits into 2 centrosomes, each with a pair of centrioles. The nuclear envelope breaks down and a spindle apparatus forms. During metaphase 2, each of the daughter cells completes the formation of a spindle apparatus. The non-genetically identical chromatids line on the metaphase plate. The nuclear membrane is completely gone at this point. For each chromosome, the kinetochores of the sister chromatids face the opposite poles, and each is attached to a kinetochore microtubule coming from that pole. During anaphase 2, the centromeres separate, and the 2 chromatids of each chromosome move to opposite poles on the spindle. The separated chromatids are now called chromosomes. During telophase 2, the chromosomes arrive at opposite poles and begin to decondense, and nuclear envelopes form around the chromosomes. Cytokinesis separates the 2 cells into 4 unique haploid cells. At this point, the newly formed nuclei are both haploid. The nuclear envelope is fully intact. The cells produced are genetically unique because of the random assortment of paternal and maternal homologs, and because of the recombination of maternal and paternal segments of chromosomes that occur during crossover. As a result, at the end of meiosis 2, either 4 sperm cells or 1 egg is produced. Our video ends with 4 sperm cells.